This recipe provides one method to identify differentially expressed genes from RNA-seq read data. With the example data provided, the goal is to validate a Saccharomyces cerevisiae knockout of the SFP1/YLR43W gene using 3' digital expression RNA-seq reads. We do this by first aligning the reads, estimating expression, and then visualizing the alignments. We start by creating a subdirectory within our home folder by clicking the small arrow and hitting Create Subdirectory. We name the subdirectory diff-RNA-seq. To begin, we need to get gene annotations from the UCC table browser. As a side note, you can use your own annotations as well. Here we select Other, then Saccharomyces cerevisiae, SAC3, Genes and Gene Prediction Tracks, SGD genes, SGD gene for the table. We want it for the entire genome and no filters, etc. We want GTF output and we will name it SAC3 SGD. We also want to select the GenomeSpace button and hit Get Output. This will send the output to GenomeSpace. We now go back to GenomeSpace, and as you can see here, the GTF file is in our home directory. We now want to align the raw sequencing reads to the genome by using top hat in GenePattern. We first go to the module, and then we select the genome, Saccharomyces cerevisiae, and we fetch the raw sequencing reads from public, shared data, demos, analysis recipe 2, and we select a wild type and send to reads pair 1. We also take the GTF file and send to GTF file. This will automatically fill in the parameters. We also do not want to find novel junctions. The rest of the parameters can be left default for this example. We now hit the Run button and send the job to GenePattern. We now want to align the mutant reads to the genome as well. We do this by hitting the little arrow, Reload. This will reload the job with the same parameters. As a side note, you can do this entire analysis with paired end data as well by feeding to read pairs 2. We now go to the same folder as before and find the mutant reads and send these mutant reads to read pairs 1 by hitting the arrow and send to read pairs 1. We then hit Run. After a brief period, this will finish and we will see these results. We now want to find what genes are differentially expressed. We do this by sending the first BAM file to cuffdiff, and this will automatically fill in the first BAM file in the first parameter, as you can see here. We now send the second BAM file and send it to the second BAM file, which will then fill it in there. So we also want to go get the GTF file and send it to the GTF file parameter. The other parameters are specific to your data and you should look at them if you need to. The other parameter we want to change is min alignment count to 0, and we want to do first read unstranded for this specific data set. And then we hit Run. This generates genes.fpkm.tracking. This contains our expression results. We also want to view the alignments to the genome. We do this by first indexing each of the BAM files from top hat. This is done by sending to Picard sort SAM. We send the first one, and as you can see here, we need to put output format to BAM and hit Run. While this one's running, we want to go back to the initial home page and send the second mutant alignment to sort SAM as well. We do this by clicking the arrow and then sending to Picard sort SAM as well. We again pick BAM as the output and then we hit Run. Now that it's finished, we want to save both of the BAM files back to GenomeSpace. Click the arrow, save to GenomeSpace, send them to your folder you created earlier. We do this for the BAM alignment file, which actually contains the alignments, as well as the index file, the .bai file. After sending it to GenomeSpace, we want to go back and send the mutant alignment to GenomeSpace as well. We do the same procedure and the same folder. After we're done saving, we now go back to the home screen and we want to save a couple of the cufflinks output to GenomeSpace. Specifically, we want to save isoform tracking and genes tracking to GenomeSpace. These are the files that contain the actual expression results. We also want to convert the cufflinks output FPKM tracking to GCT format, which GenePattern can understand. We do this by sending to FPKM tracking to GCT module, selecting tracking ID, locus, and yes. We then hit run. After the conversion is done, we save the GCT file back to GenomeSpace in the folder we created. Now back in GenomeSpace, we want to send both alignment BAM files, which have been indexed, to IGV by selecting them and then launching IGV. This will now launch IGV hosted on your own personal computer. Now in IGV, you can see that both BAM files have been loaded, but we also want to load the expression results that are in GCT format. We browse and select it from the GenomeSpace file menu. Now we go to the knockout gene, YLR403W, and hit enter. As you can see here, the wild type has the reads aligning at the beginning of the gene, while the mutant does not. As you can see, the expression values mirror this as well.